Welcome everyone. As everyone is populating into the webinar, I would like to get us started. Welcome to the Global Virus Network's Forefront of Virology webinar series. I'm Dr. Maggie Lynn Bartlett, the Program Director for the GVN and your moderator today. We are so pleased to have Dr. Hongshuo Song with us to present Escape by Shifting, Viral Entry Pathway Alteration as an Evolutionary Mechanism of Immune Evasion. Dr. Song is an associate professor at the University of South Florida, and her lab is focused on fundamental aspects of HIV molecular virology, transmissibility, and immunopathogenesis. She holds a medical degree and a Ph.D. from Peking University and completed her postdoctoral work at Duke University Medical Center. She has been lauded with several Young Investigator Awards, as well as published in top-tier journals from Nature Communications to PNAS. And we're very excited to have her today to enlighten us on this intriguing topic. Dr. Song, please take it away.
Thank you so much, Maggie, for this introduction. So good afternoon, everyone. It is my great pleasure today to share with all of you for this GVN webinar. So my lab focuses on HIV virology. So today, the topics we focus today is about a concept we are working on about virus entry pathway involution. So we originally proposed this concept based on our observation of HIV co-receptor switch. However, we think this could be a general concept, have a general important application to different viruses rather than only important for HIV. Okay, so let's start from a big picture. So why we study this topic, why we think this is important. So as most of you know, so virus entry pathways play a central role for virus cellular and tissue tropism, transmissibility, pathogenesis, and even host range. So virus entry pathways largely governed by virus receptor, co-receptor usage. An alteration of virus entry pathway can have a significant impact on virus pathogenesis and the host range. An example is HIV co-receptor switch, which is strongly correlated with disease progression in HIV-infected people. And other examples, like we are familiar with coronavirus, influenza virus, alteration virus entry pathway receptor co-receptor usage can cause a zoonotic potential, even cause a pandemic. So I will give a little bit more background of HIV co-receptor switch. I think as most of you probably already know, so for some reason that is still not completely clear, the vast majority of the transmitted founder HIV use CCR5 as its co-receptor. However, in 50% of HIV-infected people, so another kind of virus which use CXCR4 as a co-receptor can emerge after about 5 years of infection. It is important to point out here, so the 50% of 5 years is largely based on our study on subtype B HIV. So right now, we know better that for some other subtype, like subtype D and CRF01AE HIV, so co-receptor switch can have a, it can be much higher than 50% and happen much earlier during infection. So why we are interested about this phenomenon? That is because the co-receptor switch is strongly correlated with faster CD4 loss and rapid progression to AIDS. However, for many years, the molecular mechanism for co-receptor switch is it's a long-standing mystery in the HIV field. There are a couple of hypotheses. So currently, the dominant hypothesis is a target cell hypothesis. So it's a hypothesis that the driving force of co-receptor switch is the depletion of CCR5 target cells during the infection. However, the problem is that if we do a comprehensive literature search, so there's no data, especially in vivo data, can demonstrate the target cell hypothesis is true. So actually, HIV is not just a particular case. As right now we have more and more knowledge on virus receptor, it become pretty apparent that actually many viruses, they have a entry pathway flexibility. I'm just listing some examples here, like HIV, SIV, of course, and coronavirus, influenza, hepatitis C virus. But if we read the literature and the textbook, there are many more examples of this. So largely due to the high mutation rate, high recombination rate, or in other words, the high level of genetic flexibility, especially for the RNA virus. So virus entry pathway can change through minimal number of genetic alteration. So just like sometime a single mutation is sufficient to trigger this alteration. So although the virus entry pathway have a significant impact on virus pathogenesis host range, currently a fundamental gap in our knowledge is what are exactly the natural force driving viral entry pathway alteration in nature. So most of our knowledge about this is obtained from just based on laboratory setting using reverse genetic mutagenesis. However, the problem is that in nature, this process is difficult to be visualized. So the central hypothesis of the escape by shifting is that for virus with the entry pathway flexibility, entry pathway alteration, they can function as an evolutionary mechanism of immune evasion in natural infection. So in other words, the host immune responses, particularly the antibody response, can function as a driver to trigger this process. So this hypothesis or concept establish a link between the molecular mechanism of viral entry pathway, involution, and the immune evasion. So in today's webinar, we will focus on two real-world examples. The first one, our major focus will be the phenomenon of HIV co-receptor switch. And I will briefly mention the emergence of SARS-CoV-2 Omicron variant as another example. So actually, this concept came to my mind about 10 years ago when I was working in China CDC. So in this study we published in 2019, so we observe an unusually high frequency of X4 virus during relatively early infection stage. So this observation, it's the people we studied is all about within 2 years of HIV transmission. So basically, this observation they challenge the current predominant hypothesis or the dogma that HIV co-receptor switch is simply an event happened during late stage of infection due to the target cell availability. So a very interesting observation in this study is that we found a genetic signature in all the X4 virus isolated from HIV-infected people. This mutation, as you can see, located in the V3N301 glycan site. So in later study, we confirmed the importance of this mutation because as I will talk later, we demonstrate this mutation during natural HIV infection. It can function as a driver mutation to trigger the co-receptor switch of the founder virus. Another important point to mention is that this N301 glycan is located in a very important neutralizing epitope targeted by V3 glycan neutralizing antibodies. A study published in Science Translational Medicine in 2017 studied the co-evolution of HIV and autologous neutralization responses. So they found that during natural infection, HIV can evade autologous V3 glycan neutralizing antibody response by dropping this glycan. So this mutation is exactly the same location as we observed in the X4 virus. And another important evidence based on epidemiology study, so the V3 glycan neutralizing antibody have a relatively high prevalence in HIV-infected people in comparison to other antibody responses. So all this evidence actually they indicate a potential link between co-receptor switch and immune escape. So in collaboration with the US military HIV research program, so we are very lucky that we can able to better understand the process of co-receptor switch by focusing on patients from acute infection cohort called RV217 cohort. So in the study we published in 2023 in Biomedicine, so we followed up the longitudinal evolution of the R5 tropic founder virus for about 3 years. So we tracked how the virus evolved at a single genome level in order to capture the earliest time point of co-receptor switch. So we found that the earliest X4 virus, or we call it here the founder X4 virus, it emerged after about 2 years of HIV transmission. So the mutation responsible for the co-receptor switch, we call it driver mutation, again located in the V3N3 glycan site, as we mentioned earlier. So a more detailed characterization of the founder X4 virus shows that they have different mutations across the envelope compared to the founder R5 virus. However, only the mutations in the V3N301 glycan site is responsible for co-receptor switch. Another very interesting observation is that the founder X4 virus have impaired CCR5 usage. It can use CXCR4 very strong, but its ability to use CCR5 is almost completely damaged. So this is an example of another participant we studied. So different from the first one, for this participant, the driver mutation responsible for co-receptor switch located in the V3 chroin. So the V3 chroin again is an important neutralizing antigenetic epitope in HIV genome. So again, we see the founder X4 virus have impaired CCR5 usage. So what happened after co-receptor switch? What happened for the in vivo viral population? So actually, we observe a very obvious divergence in terms of a CD4 subset targeting after the emergence of the X4 virus. So by a phylogenetic analysis, it's very obvious that the founder X4 virus are replicating in the central memory and the naive CD4 subset. So these two subsets, they express high level of CXCR4, but a relatively low level of CCR5. So in the meantime, the coexisting R5 virus are replicating in the effector and transitional CD4 memory subset, which express high level of CCR5 but low level of CXCR4. So there are two important points I think we need to mention. So actually, after co-receptor switch, the CCR5 virus still the predominant virus in plasma. So while the CXCR4 virus is a minor population, it's a minor variant. This phenomenon is largely due to the target cell, because as we know, the target cell of CCR5, the effector and transitional memory, they are at a more activated status, so they can release more virus in plasma. So normally we call this as a burst size. So another important point to mention is we did not observe a depletion of the R5 target cells either before or after co-receptor switch. So this basically, again, indicate the target cell hypothesis cannot explain the phenomenon of co-receptor switch. So we look at the timing of the emergence of the X4 virus together with the dynamics of autologous neutralization activity. So very interestingly, we see that in both participants, the earliest X4 virus, the founder X4 virus, they emerge when the autologous neutralization activity increasing to the peak. And both virus, the founder X4 virus from both participants, they can completely escape their autologous neutralization. So we further looked at the virus sensitivity to a panel of very well characterized broadly neutralizing antibody, or we call it BNAbs, targeting different HIV envelope domains. So it's very interesting. The founder X4 virus completely resistant to all the V3 glycan BNAb we tested. And when we introduce the driver mutation to the founder virus genome, so this two mutation is sufficient to cause a completely resistant to V3 BNAbs. And when we revert this driver mutation to the wild type, to the founder virus sequence, the neutralization sensitivity of the founder X4 virus is fully restored. So this observation basically demonstrates that driver mutation is responsible for both co-receptor switch and immune escape to the V3 BNAbs. So actually all the X4 virus we studied so far is all completely escape V3 glycan BNAbs. So they are universally resistant without any exception. So actually, all this evidence, they kind of point to a possibility that HIV co-receptor switch is a particular evolutionary route of immune evasion. So we can summarize the evidence we have so far. So we see that driver mutation was selected at the peak of autologous neutralization activity. They can confer complete escape to autologous plasma and V3 bNAbs. And the founder X4 virus have impaired CCR5 usage. And another interesting and important evidence is that actually recent structure study, they show that the footprint of V3 bNAbs overlap with the CCR5 binding region of HIV envelope. So actually when the footprint of a neutralizing antibody overlap with virus receptor or co-receptor binding domain, so we normally consider the virus cannot easily escape, or they must maintain their receptor usage. So actually I think there are good examples of this. For example, some we see in influenza, a phenomenon we call it antigenic drift. So during the antigenic drift, yes, the virus, they can escape the neutralizing antibody response by maintaining their original entry route or receptor usage. However, for virus, they have a receptor co-receptor flexibility. A largely unexplored possibility is that the virus can achieve immune escape by altering the receptor co-receptor they use. So we call this process the escape by shifting. So actually, the overlap between virus receptor binding site and antigenic epitope is very widely observed phenomenon for diverse viral families. So this is a very interesting phenomenon. But if we look at the relatively recent version of virology textbook, for almost every single virus, a well-studied virus, you can see, so their receptor binding domain is normally also important antigenic epitope. So I think this is a very interesting phenomenon that we certainly need to understand better, especially from a structure perspective. So you may ask, so if this is a widely observed phenomenon, do we have any evidence from other viruses for this escape by shifting phenomenon? Yes, so a comprehensive literature search identified 2 studies so far, both are in vitro studies. So the first one is a PNAS paper published in 1997. So in this study, the FMDV is passaged under the selection pressure of monoclonal antibody. So the escape variant, they drop or dispense a highly conserved RGD motif, which is necessary for the integrin recognition, the major receptor. The escape variant, they gained integrin-independent entry ability through unknown receptor. So another study is on the polyoma virus. So in this study, similarly, when the virus passaged under the selection pressure in vitro, the escape virus, they gained enhanced neurotropism. But unfortunately, the molecular mechanism of receptor co-receptor responsible for this change are not studied. So based on all this evidence, we proposed a more general conceptual framework which could be applied for different viruses with receptor, co-receptor, or entry pathway flexibility. So based on the rationales we already discussed in previous slides. So in this conceptual framework, so we use the term entry tropism space to describe all the possible entry pathways as determined by various receptor, co-receptor, or cellular factors. So during the inter-host viral evolution, the virus it explores its sequence space and the fitness landscape. So it is possible that the escape mutant, they lose their original receptor usage. And in this case, it will fall to the lowest point of the fitness landscape and undergo purifying selection. In other words, it will die, it won't be observed. But another possibility is that while during the immune escape, a virus' original receptor usage is damaged. However, in the meantime, they gained a novel receptor usage ability. So in this case, the virus can achieve immune escape by altering the receptor they use. And they may find a novel fitness peak in another different environment. It can be a different tissue or cell subset. So just as we observed in the HIV co-receptor switch, the X4 virus, after they switched co-receptor, they migrate, they shifted to the central membrane naive CD4 subset. So let's go one step further and look at what exactly could be the molecular mechanism. So during this shifting, what could be the mutational mechanism? So based on the evidence we have so far, we think there are two possible mutational pathways. The first pathway, we call it direct pathway. So in this case, the immune escape mutation itself can confer a novel entry pathway. So in other words, in this case, the immune escape mutation, they have a dual function. But another pathway, we call it indirect pathway. So in this case, an immune escape mutation, they can damage the original receptor or co-receptor usage, but they cannot confer a novel entry ability. So in this case, the immune escape mutation needs to work together with another kind of mutation. We normally call this compensatory mutation, which can confer a novel entry ability. So in this pathway, the immune escape mutation and the compensatory mutation, they work together through called the epistasis in the genetics. So by the interaction with each other. So the overall consequence is that the virus, they can achieve immune escape by altering the receptor co-receptor they use. So I think the example we talked earlier about HIV co-receptor switch is a very good example of the direct pathway. So in this case, we mentioned the mutation in the V3 glycan site. They are both the driver mutation for co-receptor switch and the immune escape mutation to evade V3 glycan antibodies. So you may ask, so do we have any example of the indirect pathway? So I think yes. I think the emergence of the SARS-CoV-2 Omicron variant could be an example. So in 2022, there are two papers published in Nature. So both studies actually they demonstrate the Omicron variant is an immune escape variant with an altered entry tropism or entry pathway. So as we know, so the wild type SARS-CoV-2, they use ACE2 and a cellular factor called TMPRSS2 to enter the target cells. However, the Omicron variant are different. So they have a impaired S1, S2 cleavage ability, and also impaired usage of the TMPRSS2 enzyme. So however, it gained an enhanced endosomal entry pathway. So most likely due to this altered entry pathway, the Omicron variant showed altered cellular tropism toward the upper airway cells. So there are a total of 36 mutations in the spike of the Omicron variant. So in the N-terminal domain, receptor binding domain, as well as S1, S2 cleavage site. However, so far, I don't think the function of this individual mutation as well as their interaction are very clear in terms of the immune escape and the entry pathway alteration. So the study published in Nature Microbiology in 2022. So in this study, the author, they performed a domain swap or exchange experiment. So very interesting, they found that Omicron receptor binding domain is lethal in the wild type virus background. So in other words, if we only put the RBD of the Omicron to the wild type virus, the virus won't be able to survive. This is largely due to it can impair S1, S2 cleavage and impair the TMPRSS2 usage. But very interesting, the mutation located in S2 domain can confer an endosomal entry pathway. So however, as I mentioned earlier, so the exact mutations, this experiment is at a domain level, not at mutation level. So it's not exactly clear which mutation is doing what here. So we hypothesis the altered entry pathway of the Omicron variant is actually a consequence of the immune evasion, the escape by shifting process through the indirect pathway. So in this case, the immune escape mutation in the RBD, they impair TMPRSS2 usage. So that's the reason why when we put the RBD in the wild type virus, it's lethal. However, the compensatory mutation in S2, they provide alternative entry pathway through the endosomal. So the overall consequence, as we observe, is the Omicron variant. It's an immune escape variant with an altered entry pathway. But of course, we need to further demonstrate this by looking at the function of each mutations. So I think I will conclude the webinar today by just briefly summarize the potential implications of the escape by shifting concept in general virology. So in terms of fundamental virology, we think it reveals a previously uncharacterized mechanism of immune evasion. It also established a link between the molecular mechanism of viral entry pathway alteration and immune escape. It also kind of uncovered a mechanism underlying the origin of novel entry phenotype, which potentially can alter virus host range and even causing pandemic potential. In terms of the implications for the future vaccine therapeutic development, so due to the time today, I don't think, because they're in terms of co-receptor switch, because HIV is a very, it's a, HIV itself, I think it's a big field. So there could be many implications in terms of co-receptor switch. But today I just want to briefly mention in a general sense. So what could we learn from this for the future vaccine therapeutic development in general? So I think one important point is it's probably very important to know for any particular virus, we need to know its entry tropism space, which means all the possible entry pathways it can explore. So we can so if we know better of the entry tropism space, we can possibly develop a novel therapeutic or vaccine strategy to block all possible entry pathways instead of only focusing on a single pathway. So I think one example is HIV. For example, the V3 glycan BNAb, they work very well. They are very broad in terms of CCR5 virus. But for the X4 virus, it's almost completely resistant to this class of BNAbs. So I think if we future vaccine, so if we can really develop some strategy that can targeting the entire entry tropism space, it is possible that we can develop some really broad strategies to prevent virus immune escape. So there are many people that I need to thank today, and I will I hope to show particular thanks to our institute director, Dr. Gallo, who gave a lot of support in the past few years about this study, and also many collaborators. I won't be able to list all of them. Yeah, I will be happy to take questions. Thank you.
Thank you, Dr. Song. That was a fantastic presentation. I would like to direct people to put their questions in the Q&A box, and I'll go ahead and moderate those for you, Dr. Song.
Okay, thank you so much, Maggie.
Yes, so Sten Vermund, the Chief Medical Officer for GVN and Dean at the College of Public Health at USF, is asking, can you discuss further how monoclonal antibodies that target part or all of the co-receptors might work well?
Oh yeah, I think this is a very, very important question, of course. But the thing is, I think we can probably achieve this by engineering the current existing antibody. But I'm not very sure about whether in natural HIV infection so far there are any broadly neutralizing antibody targeting both CCR5 CX. But of course, the CD4 for HIV, I think the CD4 binding site antibody it's a because I don't think so far we know HIV that does not use CD4 yet. Yeah.
And further, how conserved are these co-receptors across circulating variants? And then, Dr. Song, your camera is off, just so you know. So if you could turn it on, that'd be great. Yes, now I can see you. Great.
Thank you. I just turned it off. I'm sorry, what's the next one?
Oh, how conserved are the co-receptors that you're identifying across different variants?
Oh, yeah, actually the prevalence of the co-receptor X4, I think is highly depend on HIV subtype. As we mentioned, the most extensively studied subtype is in the old days, subtype B. It's about 50% after 5 years of infection. But in subtype D, so for example, prevalent in Uganda and some African countries, there are certainly high prevalence. But I think one question still controversial is the subtype C virus. There are studies they show the subtype C probably never undergo co-receptor switch. Yeah, but there are some controversy, some literature they say, okay, there are a lot of X4 for subtype C, and some study they does not observe. I think my opinion is this, because even for a single HIV subtype, there are also different phylogenetic clusters. So my opinion for subtype C is probably depend on the exact clusters. Yeah.
Yeah, that's fascinating. Margaret Brosnahan asks, if HIV can use multiple entry routes, how come people who don't express CCR5 are immune to infection?
Oh yeah, I mean the CCR5, right? The
Yes.
CCR5, yeah. I think for the in the there are studies the people defective the have the delta 32 allele they are defective for the surface expression CCR5. But the people they can still obtain HIV infection. So people characterize the virus. It turn out to be either UCXCR4 or other some uncommon co-receptors. But I think this another project actually we are currently working on. I think the reason why R5 virus is more transmissible is not because R5 itself, it's because the cells in the mucosal site, they express high level CCR5 but low level CXCR4. Yeah, so because the target cell for CXCR4 is really all located in the lymph node because they are central memory naive. They are not that much located in the mucosal site. Yeah. So I think this is another things we are currently working on right now. Yeah.
Thank you. I wonder how your findings today change perhaps the clinic might approach these infections or thinking about new treatment modalities.
Yeah, I think one important in terms of HIV field, so there are a couple of important points. I don't mention that very detailed in this webinar, but I think one thing is we need to probably be careful about the using the therapeutic approach using only a single neutralizing antibody. So there is a risk that we can select for the X4 virus during the treatment if we use that as a functional cure or prevention strategy. Another thing I think it's clinically important. It's actually based on our study. So all the X4 virus, in plasma, they exist as minor variant. So sometimes even using the deep sequencing, you are not able to detect the X4 virus. But we see that the patient, they have an extremely low CD4. But when we take the plasma, we sequencing or we even look at the functional phenotype. So sometimes we cannot even detect X4. But when we look at the virus in the cells, especially in the central memory and naive subset, we see, oh, there are all X4 virus there. So I think this is, in my opinion, this is a real concern. So in the future, I think if we screen the people, okay, whether you have X4 or not, we need to use the cell instead of plasma. So again, I think this is determined by the nature of the target cells, the CXCR4 virus, because they target in the naive and the central memory. So these two kind of cell subset, they are relatively resting in vivo. They won't release that much virus. Yeah. But they still pathogenesis wise, it's still significant. Because as we know, the HIV, they can kill the resting cells by different mechanisms. Yeah. It's not necessarily only the virus predominantly in plasma is important. Yeah. I think this is a real concern.
That's such an important point.
Thank you.
So Michael Martin asks, in other viruses, do these co-receptor variations have significant effects on tissue tropism?
Oh, I think I actually for other virus I don't look at that details, but I think yes. But so actually the concept of our concept is not only the receptor co-receptor, but like as we see in the Omicron variant, it's a generalized, it's an entry pathway. So actually the virus entry pathway, they are very complicated. So I think this is not very technically straightforward. That's the reason why. So we cannot using a very quickly a lab method to determine all the receptor. It's time consuming, yeah. So, but I think yes, certainly. I think different tissue organs they have different requirement. So we call this entry barrier. For example, HIV, we show that, and also the the Omicron, they kind of more targeting upper airway. So they certainly a, a receptor or co-receptor change, they can change the target cell specifically. Yes, yes.
Thank you. And could this immune evasion for SARS-CoV-2 somehow link to long COVID or prevalence of SARS-CoV-2 reservoirs in post-acute infections?
Oh, I'm not a COVID expert, so this is a difficult question. Yeah, but I think yes, it have implications. Yeah, so because we don't know the SARS-CoV-2, they probably still have other potential that we don't know yet. So technically, I still don't know what's the best way we can determine all the possible entry pathway receptor co-receptor, because different cells, different tissue organ, they are certainly different. Yeah.
And I wonder if you might talk a little bit about your experience as a researcher on HIV who then started working a little on SARS-CoV-2 during the pandemic, and how that has enriched your approach back to HIV, or how you see
Oh.
the research landscape now.
Yeah, actually, I always focus on HIV. Actually, our lab does not do very much about the SARS-CoV-2. We just interest, we just look at the sequence, we just look at the literature. But in terms of the real experiment, actually, we always focus on HIV. So we are interested about the COVID, just like the Omicron, the origin of Omicron, evolutionary origin, but we have not got any chance to do any actual work yet. So it's difficult to focus on very deeply on two different things. Yeah.
And as far as the tropism and different receptors, you talked a lot about what's been known for humans. Has other, like SIV and the receptors, been well characterized in non-human primates or other species
Oh
that are
yeah,
part of the pathway?
this is a very important question, actually. I think this is certainly another seminar deserved.
Yeah.
Yeah, actually, in the old days, people think that SIV, they also use CCR5 in their natural host. But there are some research, it's not very recent, I think 10 years ago. So they actually say that SIV, just like SIV, HIV, the core receptor use is complicated. For example, they can use CXCR6 and FPRI1, a lot of the 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 structural similar things. I think there is an opinion. It's also what we are very interested about. It's a SIV in their natural host probably does not use CCR5 as their major transmission co-receptor. So that's the reason why, because I think 50% of the natural hosts, like the sutimagavir, they are defective in terms of CCR5 expression. But in these populations, animals, they obtained SIV infection at similar frequency percentage as the animal who has a wild type CCR5. So I think this is just there is I think I believe this is true. So the SIV probably does in their natural host, they won't use CCR5 as a major co-receptor. They probably use something else, but we are not very clear. Yeah.
No, that's very fascinating. I wonder if you might also elaborate on the broadly neutralizing antibody treatment avenue, in order to avoid pushing escape mutants. Is there an ideal number of BNAbs to use in a therapeutic? Has that been elucidated, or?
So I think a combination of different BNAbs, this is a concept of the entry space, actually. For example, if we combine the CD4 binding site and the V3 glycan, probably something else targeting different envelope domains. I think as long as you can block all possibilities, there will be no room for the virus to escape. Yeah. I think we can certainly achieve that. It's just we don't know how, because we need to certainly understand better about the virus entry mechanisms.
Do you anticipate there'll be additional receptors that are found as
Oh, you mean, yeah, I think for HIV, the two major co-receptor are clinically relevant is CCR5 and CXCR4. But actually in vitro, because we do a lot of the in vitro work. So the virus, they can use many other things like CCR3 and APG. And actually, this probably not that clinically relevant because if we use PBMC, we find that if we use a CCR5 inhibitor, CXCR4 inhibitor, even some virus they can in the cell line they can use some other something else. They won't be able to enter the PBMC very efficient. But I think yes, if we look at the macrophage, different target cells, different organs, it's probably different. Yeah. I think in the old days there are also some report that some HIV cannot don't need CD4. Yeah.
Right. No, this was fascinating. Thank you so much, Dr. Sung, for your intriguing webinar, and thank you for everyone who's asked questions. I want to remind you that we will have another Forefront of Virology webinar on October 23rd at 11 a.m. Eastern Time. Continuing medical education accreditation for that is pending. You can submit for CME for this if you message Ruby. And the talk next month will be on molecular mimicry as a strategy for anti-cancer immunity. Thank you again, Dr. Sung, and we will see you all next month.
Thank you so much, Maggie. Thank you, everyone. See you.